Welcome back, my dear students. Today we are going to explore how the delicate balance of ions across the cell membrane creates the resting potential and how a sudden change in this balance sparks an action potential, the very basis of nerve communication. Step by step, we will see how these impulses travel along axons, sometimes slowly and sometimes in rapid jumps, ensuring that messages reach their destination with perfect accuracy. By the end of this class, you will clearly understand how tiny shifts in voltage gives rise to the incredible electrical language of our nervous system. Resting membrane potential, or RMP. Basically or fundamentally, the resting membrane potential is the electrical voltage differences across the membrane of an excitable cell like neurons and muscle cells. It exists because of unequal ion distribution between the inside and outside of the cell. Resting potential is basically the difference in electrical potential across the plasma membrane when the cell is not stimulated or when the cell is in a state of relaxation. Key factors responsible for resting membrane potential, what is minus 70 millivolt in neurons. Unequal ion distribution, extracellular fluid or ECF, what is that is rich in Na plus and Cl minus, cytosol inside which is rich in K plus. Major anions are phosphate groups, just like ATP, and negatively charged proteins. Membranes has more K plus leak channels than Na plus leak channels. More K plus leaves than Na plus enters inside, becomes negative. Inability of most anions to leave. Large negatively charged molecules like ATP and proteins cannot cross the membrane. These trapped anions add to the negative charge inside. The third reason is the electrogenic Na plus or K plus pump. That is Na plus K plus ATPase. It pumps 3 Na plus out and 2 potassium plus in. It removes more positive charges than it brings in. It contributes slightly about minus 3 millivolt to the overall resting membrane potential, and it maintains low Na plus inside and high K plus action potential or AP. An electrical signal or nerve impulse that travels along the action's membrane is the action potential. It is caused by movement of Na plus and K plus ions through voltage gated channel. Once triggered, it always propagates with the same strength, that is all or none principle. This is the image showing action potential. This is the electrical signal that travels along neurons membrane. This is the resting membrane potential. This is the phase of depolarization. This is the phase of repolarization, and it is hyperpolarization. Now, graded potential, small local changes in the membrane potential. It can make inside of the cell either more negative or less negative. Types: hyperpolarizing graded potential, inside becomes more negative further from threshold. Depolarizing graded potential, inside becomes less negative or closer to threshold. This is graded potential. A weak stimulus causes small depolarization. A strong stimulus causes large depolarization. A stimulus of longer duration causes a long-lasting depolarization but of almost the same strength as the previous stimulus. A strong stimulus that depolarizes above the threshold triggers an action potential in the postsynaptic neuron, and stimuli that result in hyperpolarization depend on the activation of specific ion channels. Generation and propagation of action potential. An action potential, we know, is it's basically a sequence of rapid events that reverse membrane potential and it restores to resting state. The phases are depolarization, inside becomes positive; repolarization, it returned to minus 70 millivolt; and after hyperpolarization, inside more negative than minus 70 millivolt, approximately minus 90 millivolt. So what happens during resting state? All voltage gated Na plus and K plus channels are closed. Then action plasma membrane is at resting membrane potential. Small buildup of negative charges occurs along inside surface of membrane and equal buildup of positive charges along outside surface of membrane. The second phase is the depolarizing phase. When the membrane potential of action reaches the threshold, Na plus channel activation gets open. As Na plus ions move through these channels into neuron, buildup of positive charges forms along inside surface of membrane and membrane becomes depolarized. Third, repolarizing phase begins. Na plus channels inactivation gets closed and K plus channels open. Membrane starts to become repolarized as some K plus ions leave neuron and few negative charges begin to build up along inside surface of membrane. Fourth is the repolarization phase continues. K plus outflow continues as more K plus ions leave neuron, more negative charges build up along inside surface and membrane. K plus outflow eventually restores resting membrane potential. Na plus channel inactivation gets open and return to resting state occurs when K plus gets closed. Then again resting state will be occurred. This is a cyclical process. Phases of action potential: depolarizing phase triggered when membrane depolarizes to threshold, that is minus 55 millivolt. Voltage gated Na plus channels open, that is Na plus rushes in. Membrane potential changes from minus 55 millivolt to plus 30 millivolt. Positive feedback: more Na plus entry and more channels open. Repolarizing phase: Na plus channels inactivate, voltage gated K plus channels open, which is slower, and then potassium flows out, that is potential returns from plus 30 millivolt to minus 70 millivolt. After hyperpolarizing phase, K plus channels stay open longer, membrane becomes more negative, that is approximately minus 70 millivolt, and it returns to resting potential as potassium channels close. So what is the all or none principle? Action potential occurs only if threshold is reached. Subthreshold stimulus will cause no action, no generation of action potential. So during at subthreshold stimulus, no action potential will be generated. So threshold stimulus is approximately minus 50 millivolt, which causes one action potential, and suprathreshold stimulus multiple action potentials but some amplitude, not stronger, just more frequent. Analogy: domino effect, either all fall or none fall. This is the image showing propagation of action potential. It showing that previous active area returned to resting potential and adjacent area that was brought to threshold by local current flow now active at peak of action potential. New adjacent inactive area into which depolarization is spreading, it will soon reach threshold. So remainder of action still at resting membrane potential. So this is the process by which action potential propagates through the length of the action. Now we'll study about the refractory period. Refractory period is basically two types: absolute refractory period and relative refractory period. Absolute refractory period is the sodium channels active or inactivated, but no new action potential possible. It ensures one-way conduction. Relative refractory period: sodium channels reset and K plus still open, stronger than normal stimulus needed for action potential. Large actions, it is shorter refractory period, that is approximately zero point 0.4 millisecond up to 1000 impulse per second. Small actions, longer refractory period, which is approximately 4 millisecond up to 250 impulses per second. This is the image showing the refractory period, absolute refractory period and relative refractory period. Absolute refractory period, we know it happens due to inactivated Na plus channels, and relative refractory period occurs due to continued outward diffusion of K plus channels. Propagation of action potential. Action potential travel from action hillock, that is the trigger zone, to action terminals, we know it. Unlike graded potential, action potential does not decrease in strength, that is not decimetral. Propagation relies on positive feedback. Types of conduction: continuous conduction, step by step depolarization and repolarization. It occurs in unmyelinated action and muscle fibers, and it is a slower process, that is the continuous conduction. But saltatory conduction, it action potential jumps from node to node, nodes of Ranvier, myelin sheath. There is no ion flow at covered region. It is much faster than energy efficient. Fewer Na plus or K plus pumps needed. Factors affecting speed of conduction: there is a myelination. Myelinated actions conduct faster. Action diameter: larger actions show faster conduction, and temperature: cooling shows conduction speed. So in today's class we uncovered how the resting membrane potential sets the stage for excitability and how action potentials are generated, conducted and propagated to transmit signals with accuracy and speed. We saw how ion movements, threshold, refractive periods and type of conduction work together to make nerve impulses possible. The beautiful orchestration is what allows our nervous system to function like a high-speed communication network, controlling everything from reflexes to reasoning. So always remember, every thought, movement and response begins with a tiny change in membrane potential. So keep practicing, stay motivated, and I will see you in the next class. Thank you.